Welcome back to MicroVerse. In the previous lecture, we explored the bacterial cell wall, cell membrane, and cytoplasmic structure. Now we move to an important bacterial appendage. Some bacteria can actively move from one place to another. But how? The answer is bacterial flagella. These are the principal organ of locomotion in many bacteria. But today's question is not simple. What is flagellum? Instead, we will understand how this bacterial flagellum arranged structure and how does it produce movement. So let's see what is flagellum. A bacterial flagellum is a long, thin, unbranched filament that acts as an organ of locomotion. Not all bacteria possess flagella. The presence, absence, number, and arrangement of a flagella may be characteristics of particular bacterial groups and can help in their identification. So let's see the arrangement of a flagella. So first look at how flagella may be distributed over the bacterial cell. First, monotrichous. Mono means one. A single flagellum is present at one pole. For example, Vibrio cholera. Then Lophotrichus means a tuft of flagella is present at one pole. Example is Spirillum. Then Amphitrichus. In this, flagella are present at both poles. Example, A. Vigalis. Then the peritrichus. Flagella are distributed all around the bacterial cell. Example is typhoid bacilli or salmonella. Then the structure of the bacterial flagellum. Now comes the most important part. Every bacterial flagellum has three major parts: filament, hook, and basal body. But each of these has a specific structure and function. First, filament. The filament is the long external part of the flagellum, which is hollow, rigid cylinder extending from the cell surface to the tip. It is composed of the protein flagellin. Flagella of different bacteria may be antigenically different. These flagellar antigens are known as H antigen. They induce specific antibodies and are useful in serodiagnosis. So remember, the filament is made up of protein called flagellin. Because flagellin can act as an antigen and it is known as the flagellar or H antigen. Then, so remember, the filament is made of flagellin and flagellin gives rise to the H antigen. Then second part is hook. The hook is short curved structure located between the filament and the basal body. Its function is to connect the filament to the basal body. You can understand it is a flexible coupling that allows movement to be transmitted from the basal region to the external filament. Then the basal body. The basal body is the part embedded within the bacterial cell envelope. It consists of a central rod and a number of rings. This, this is where the gram positive and gram negative bacterial structure differ. So let's see the basal body in gram negative bacteria. Classically, gram negative bacteria have four rings: L, P, S, and M. Let's understand their locations. So first L ring, the outermost ring, it is associated with the lipopolysaccharide layer or outer membrane. Then the P ring, which is located at the peptidoglycan layer. Then S ring, which is located just above the cytoplasmic membrane. At last, M ring, which is associated with the cytoplasmic membrane. So the classical arrangement is L, P, S, and M from outside towards the cytoplasmic membrane. Then the basal body in gram positive bacteria. Gram positive bacteria do not possess an outer membrane, therefore, the basal body is structurally simpler. The P and L ring are present. The basal body has the inner membrane associated and outer supporting components corresponding to its simpler cell envelope architecture. Then the important concept, the central rod. All these rings are connected by a central rod. The rod acts as the central structural axis of the basal body. The external filament is connected to the basal structure through the hook. So visualize the complete flagellum as a basal body, hook, and filament. Then how does the flagellum produce movement? The bacterial flagellum does not move by whip-like blending action like a eukaryotic flagellum. Instead, the filament rotates because the filament is rigid and helical. Its rotation works like the propeller of a boat. The rotational movement pushes the bacterium through the surrounding fluid. Therefore, a rotating flagellum is equal to bacterial locomotion. The direction of rotation influences the pattern of movement. In peritrichous bacteria, coordinate rotation allows flagella to form a bundle for forward movement, while a change in a rotation direction can disrupt this arrangement and produce tumbling. So next point, where does the energy come from? The flagellar motor is associated with the basal region. Energy for rotation is generally derived from proton motive force. In simple terms, the movement of protons across the bacterial membrane generate energy that derive from the flagellar motor. Some bacteria can use a sodium ion gradient instead. This energy is converted into rotational movement, then rotation of the basal apparatus, next rotation of the filament, and last bacterial movement is happen. The next point is formation of the flagellum. The flagellar formation is a highly organized process. The basal portion is assembled first, then followed by the structural external structures. A useful simplified sequence is basal body, hook, and filament. The filament grows by the addition of a flagellin subunit, which are transported through its hollow central core towards the growing end. This means that the filament grows at the tip, not at the base. This is an important concept in a flagellar assembly. The next point is importance of flagella. Flagella are important because they confer motility and produce characteristic patterns of a movement. The movement patterns may assist in a bacterial identification. For example, tumbling motility, which is seen in Listeria. Then darting motility, seen in Vibrio cholera and Campylobacter. Then swarming motility, seen in Proteus and Clostridium tetani. Then the stately motility, seen in the Clostridium. Then corkscrew type of movement, seen in spirochetes. My laboratory demonstration. Because bacterial flagella are extremely thin, they are difficult to demonstrate directly. They may be demonstrated directly by electron microscopy and spatial flagellar staining methods. Then by indirectly hanging drop preparation, dark ground microscopy, and phase contrast microscopy. This distinction is useful for laboratory examination and viva questions. Then, if you are asked, describe the structure of bacterial flagella, start with three parts. First, filament, which is hollow, rigid, made of flagellin and carries H antigen. Then, hook is short and curved, which connects filament with the basal body. Then, third, basal body, which is central rod with rings. For a gram negative bacteria, there is a LPSM L, lipopolysaccharide or outer membrane, P for peptidoglycan, S for just above cytoplasmic membrane, and M is cytoplasmic membrane. So bacterial flagella are much more than simple threads attached to the bacterial cell. Their number, arrangement, structure, and antigenic properties, also type of motility, all have microbiological importance. The key concept is the basal body anchors the flagellum, the hook connects it, and the filament rotate to produce bacterial movement. So movement helps bacteria reach the right location, but once they reach a surface or host cell, another question arises: how do bacteria attach? So in the next lecture, we will see the pila and fibri. How do bacteria attach? So thank you for the learning with MicroVerse. Let's master microbiology, one concept at a time.